Hello, everyone, and it's a pleasure to be at ICML, even if virtually this year. Today, I'm going to tell you about the work that I've been doing over the last few years on transforming drug discovery using digital biology. So, drug discovery is an interesting space because you can think of it as a tale of the glass half full and the glass half empty. This is the glass half full, where in the last 50 years, the community has emerged with therapies for diseases that up until recently were were a death sentence or a sentence to a life of pain. Of course, vaccines are very much on everyone's minds, and the incredible success of the recent vaccines, which were engineered in less than a year, is just an incredible achievement for human medicine. But vaccines is only one example. There's been therapies created for cancers that have turned metastatic disease for certain cancers into something that is curable for the first time for some subset of patients. Autoimmune diseases have had remarkable therapies, and in what is perhaps one of the greatest achievements in our understanding of the human, genomes is the set of drugs that have been created for cystic fibrosis, where the understanding of the specific genetic variants that cause the mutation that are mutations in the CFTR gene have allowed 90% of cystic fibrosis patients to live an almost normal life. So these are just a few highlight examples of where drug discovery has made a tremendous impact on the life of patients. The glass half empty is this it can be captured in this chart, which has come to be called Irum's law. Irum, being the inverse of Moore, represents an exponential decrease in productivity in pharmaceutical R&D, as opposed to the very famous Moore's law, which is an exponential increase in productivity of technology. So because of this exponential decrease, the amortized cost per approved drug is now over $2.5 billion, and that emerges from an R&D success rate which, depending on how you define the denominator, is somewhere between 5 and 10%. And it is the cost of all of those failures that adds on top of every successful drug, and that is what gets us to the price tag of $2.5 billion. So why is it that so many drugs fail? If you think about the journey that a drug makes through its many phases, from idea through various phases of discovery, preclinical work, and all the way through to a clinical trial, there are many, many forks in the road. And in each of those forks, there's maybe one or two paths that take us to success, and many that take us to futile dead ends that add to the costs and the probability of failure. So what if we could better pick at each of those junctures which path to take? Fortunately, machine learning over the last decade or more has come up with better and better ways of making predictions. And so the question that we set out to ask is, can we make better predictions on which path to take, thereby decreasing the amount of time and cost spent on all of those dead ends and increasing our probability of success? So the goal of what we do at In Sitro is to integrate machine learning together with creation and collection of very high quality data to be able to predict better which path to take. We are trying to apply this approach across multiple steps in what's called the pharmaceutical R&D value chain, starting with the very beginning, which is which targets do we go after, where we're using a combination of machine learning and statistical genetics on the one hand, and in vitro, in the lab, disease models derived from what are called human-induced pluripotent stem cells to identify targets that have validity in them for human biology. We can take those targets and use a combination of machine learning and high-throughput chemical data generation to generate new molecular designs for new compounds that we can take into the clinic. And then ultimately, we want to use machine learning to inform our clinical strategy using machine learning-defined patient segments and biomarkers for both patient selection and clinical efficacy. So these are all different places in which machine learning can potentially be used to improve the probability of success. The ability to take on these very challenging problems has been driven by the fact that we are living at the convergence of two revolutions that have occurred in parallel in two very different communities. In the left-hand panel is the revolution in the life sciences, where over the last decade or two, there's been a tremendous set of tools that have been generated that enable the creation of massive amounts of high-fidelity data about human biology and about chemistry. The most famous, of course, is DNA sequencing, which has been with us now for a little bit over 2 decades, and the ability to then sequence the human genome for many, many individuals, driving our understanding of the connection between genetics and human clinical disease. These, what are called induced pluripotent stem cells, are a more recent invention, and is the ability to take cells from any one of us, revert them to a stem cell status, and then differentiate them into neurons or cardiomyocytes or liver cells or a multitude of other cells that carry our genetics, but also the right cell type that represents potentially human disease. Technologies like CRISPR have enabled the perturbation of such cells genetically in a very precise and scalable way. And then we can measure those cells using a multitude of high content modality, ranging from super resolution microscopes to RNA sequencing and many more. We have the ability to do all of this at unprecedented scale using automation and microfluidics, as well as the ability of DNA to barcode. And that really has created a perfect storm of data production on the life sciences side. What's lacking is the ability to take those data and maximally utilize those. And that's where the machine learning comes in, because people have a limited ability to understand all of the value that is in these data sets. So I don't need to tell this community about how far machine learning has come over its life, and most notably the incredible revolution that's happened over the last decade, where today we're able to address at beyond human level performance problems that a decade ago we were able to address maybe at slightly better than random. That gap in performance between a machine and a human is there even in tasks that people are good at, like labeling natural images. That gap is considerably larger in tasks that people are not so good at, like interpreting microscopy images or a whole bunch of reads taken from a patient's cell. So how can we put together machine learning with this high content data production in order to inform our understanding of disease biology? So I don't need to tell this audience about end-to-end learning. This is a slide that I use for life science audiences. So on the left-hand side, what I think is maybe useful is the right-hand panel, which is to say that when we use end-to-end learning, we're also actually learning a representation of the data. And that representation allows us to uncover relationships between data instances that might not be readily apparent in the original labeling. So as we all know, when we create a new representation in a lower dimension than the original space, distances in this representation are quite different than distances in the original data encoding. And so objects that might be very far away from each other, like the two trucks on the left, are now much closer to each other in this new manifold that we've generated. But also even potentially classes that are labeled differently, like cars and tractors, are also close to the trucks because they share some high-level features. And that learned semantic encoding is something that maybe in natural images may or may not be as useful, but when you're trying to deconvolute biology where our understanding is so limited, can be incredibly helpful. And I'll talk about that in a couple slides. So the core problem that we set out to solve at the beginning of our journey is at the fundamental roots of drug discovery, which is what would be the clinical impact of an intervention in a human. And that is a problem that is often tackled by doing experiments in animal models, whose translatability to human is often very, very limited, because no matter how much you would do experiments in a mouse, mice are still not people. So how do we come up with a model of what the clinical impact in a human would be when we can't do experiments in human beings up until the very late stages of the clinical trial? So we are going to use our machine learning models to try and create a representation of human biology that can help us answer questions such as these. So the basic tenet of our approach is that we collect high content data from human cohorts. This can be data that is collected from a human, like histopathology data from a biopsy sample, a brain MRI, an EKG measurement. Many, many other ways of measuring human biology have emerged. We can also generate high content data from each of those individuals by collecting some of those induced pluripotent stem cells that correspond to their genetics and then assaying those cells using multiple high content modality across multiple conditions and really creating a very dense representation of the biology of that individual human. Those are the data samples on which that we place into this manifold, this embedding, and we are using strong genetic drivers of disease where the labeling of those individuals in terms of their disease burden is very clear to create anchor points or positive examples of disease state versus healthy state that allow the machine learning to anchor on an appropriate representation. Now, in this space, we have a very dense, high-content representation of those humans, as well as those anchor points allow us to cluster individuals in this manifold and create some understanding of the heterogeneity in the patient population. The reason why that's important is because we now understand that most complex disease is really not one disease. It's multiple different biological processes that drive dysfunction in often very different ways. This isn't something we now recognize and utilize extensively in precision oncology, where we no longer think of breast cancer as one disease, and we treat BRCA1 patients very differently from HER-positive patients with targeted therapeutics that really focus on the specific biological drivers for their type of disease. We haven't done that for most complex polygenic germline diseases like Alzheimer's or autism on the one hand, or diabetes and cardiovascular disease on the other. And the question is, can we identify those coherent patient populations and come up with targeted interventions that treat the specific root causes for those patients? And that's what we hope to do by uncovering the structure in this manifold. So in order to construct this manifold, we're combining two types of data. The first of those is human genetic data, where we're leveraging the fact that while we can't do experiments in a human being, each of us is an experiment of nature. Each of us is a place where Mother Nature has perturbed the genetics of of each of us and given rise to a different clinical outcome. So to collect relevant data, we're able to leverage the incredible progress in human genetic sequencing that provides us with an incredible amount of DNA sequence information. So this is a graph that demonstrates just the incredible growth in this, in terms of this data modality, where it represents on a log scale the number of human genomes that have been sequenced since the very first human genome in 2001. This is a Moore's law graph. It is on a logarithmic Y scale, and you can see that not only is the number of human genomes sequenced growing exponentially, it's actually growing twice as fast as Moore's law. So by the end of this decade, we will probably have hundreds of millions, if not billions, of genomes sequenced. Genomes are only useful up to a point. They're even much more useful if combined with phenotypic data. And fortunately, that is something that is also growing, not quite as quickly as sequencing. But this, for example, is one of my favorite resources. It's called the UK Biobank. It's one of the earliest biobanks that have been generated and is particularly well constructed in measuring many, many different clinical phenotypes, including everything from cognitive phenotypes to imaging, urinary biomarkers, blood biomarkers, multiple covariates, along, of course, with the genetics that really allow us to create that alignment between genotype and phenotype. Other biobanks are now being created in other countries besides the UK. Here in the United States, there is an All of Us effort that is intended to be 1 million patients, 1 million people, not the 500,000 that are here, and also with much greater ethnic diversity than exists in the UK Biobank. The UK is also expanding to a much larger population, and there's many others. If you take the genetics and the clinical outcomes, and you juxtapose them, that association oftentimes, in many cases, represents causality. This is a pretty reasonable first cut approximation because the genetics comes first before the clinical outcome. There's exceptions to that, but it's a pretty good starting point. So you would think that between this allows us to uncover those targets, those genetic drivers of human clinical outcome and human disease. And sure enough, there's been a tremendous amount of work over the last 12 to 15 years on what are called genome-wide association studies that take those genetic variants, align them with human clinical outcome or human traits, and thereby discover the genetic architecture of different traits, where the traits go all the way from diseases like Crohn's disease or Alzheimer's to biomarkers such as QT interval or LDL cholesterol to things that are not even disease like height or educational attainment. So you would think this would be the answer to uncovering novel drug targets, but unfortunately this is only the beginning of the journey, because each of those genetic association studies uncovers in many cases very large numbers of effects, often with small effect sizes at the population level. And so disentangling which of those to go after when each drug program costs hundreds of millions of dollars is not something that can easily be done. So what we aim to do is to really use high-content biological data to get us closer to the causal biology. This is exactly those high-content biomarker data sets that I alluded to on a few slides ago. So I'm going to give a few examples of how we've done this, exploiting both our machine learning-enabled statistical genetics efforts as well as our work on iPS cells. So one of the diseases on which we've spent the most effort is what's called NASH, or non-alcoholic steatohepatitis. NASH is a disease that is generated by what's called fatty liver disease, whose prevalence is about a quarter of the world's population and unfortunately growing rapidly because it is often a direct consequence of obesity and metabolic syndrome. NASH is what happens when fatty liver, which is characterized by the steatosis or lipid droplets in the liver cells, when the liver becomes inflamed because of those fatty droplets, the hepatocytes or liver cells die, the liver becomes inflamed, and that inflammation gives rise to scarring or fibrosis. And when you get to advanced disease, the liver becomes so scarred that it's dysfunctional, and one sees liver carcinoma or cancer. This, in fact, is now one of the most common reasons for liver transplantation in Western countries and will overtake viral hepatitis as the leading cause of liver cancer in the coming years. So in order to tackle this question, we had access to a really remarkable data set from the clinical trials of our partner Gilead. This was a clinical trial that was ultimately not successful, but was very well executed, and in particular had a liver biopsy taken for each patient at the beginning and the end of the trial. Each of those was graded very rigorously by a single central pathologist, so minimal variability in grading, and were graded using 4 scores, the fibrosis score, which is actually the FDA clinical endpoint that is looking to be modulated in this disease, but also steatosis, which is the extent of the lipid droplet accumulation, inflammation, and ballooning. With that pathologist score, we asked the question of whether one can find drivers of the fibrotic progression process. And no matter how much we tried to analyze the data, that didn't really give us a lot of information because it was a rather underpowered trial with only 550 patients. So our insight here was that we could take those data and instead of just using the pathologist scores, we could go back and dig much deeper into these images, which are 20,000 by 80,000 pixels, and come up with a much more quantitative machine learning derived notion of disease state, which in turn would be able to use, we would be able to predict the pathologist scores, but would contain a lot more information and be a lot more quantitative. And so with that more quantitative measure, we would be able to subtract, if you will, the disease state at the beginning and the end of the trial, and maybe that notion of progression would give us our understanding of what are drivers of disease. So basically, we started out with these H&E stained whole slide images. We divided them into smaller tiles. We trained convolutional neural network with an attention-based pooling mechanism that allows the model to figure out where in the image, this very large whole slide, the pathologist saw fibrosis, because the slides are labeled at the whole slide image, not at the tile level. And then the attention mechanism allowed us to attribute which tile gave rise to that label, and that was how we trained the model in a very weakly supervised way. With that embedding that was derived from this training model, we were able to show that you could predict with very high quality the pathologist scores for each of those 4 scores. You can see that the agreement that we have between the machine learned predicted scores and the pathologist scores is at or beyond the level of interpathologist agreement. And as a note, this was on a very rigorous holdout regime where we tested not only on a separate group of patients, but in fact patients that were derived from a different set of clinical sites that did the biopsy. What's also notable is that the fibrosis is typically called by a pathologist using a separate stain on top of the standard H&E stain. The machine learned model got equivalent quality just by looking at the H&E staining alone, something that pathologists are not able to do. In addition to the ability to predict pathologist scores, the machine learned scores also align considerably better with other markers of patient state and patient disease. So what you see here is on the x-axis the pathologist scores. On the y-axis you have the machine learned scores, and what you see is the correlation on the left side with blood biomarkers that correspond to disease, and on the right-hand side with RNA sequencing data from those same disease livers, and you can see that both of those correlate better with the machine learned scores than they do with the pathologist scores. But maybe most important is what we were looking to do at the very start, which is uncover genetic drivers of disease progression and regression. And those are very hard to come by because progression data sets are often very underpowered because of the need to collect longitudinal samples, as they were here. So when you do the association analysis for progression with the pathologist scores, you can see that there's very few examples of a distinct shift between, say, F3 and F4 fibrosis, and that just didn't give enough examples for genetic drivers to be picked up. In our much more quantitative scores, there's more examples of movement across fibrosis regression or progression, and that allowed us to uncover 2 genome-wide significant hits for fibrotic progression, as well as several others that are at the cusp of significance, and those are biologically very compelling and eminently prosecutable as drug targets, which is what we're currently doing. So that was one of our efforts. I mentioned earlier that we have a complementary way of collecting data that allows us to to build an intervenable platform based on the biology of individual humans. So this approach begins where our work on statistical genetics leaves off with a genetic architecture of human disease. We then create that genetic diversity in an in vitro population, a little in vitro human population in a dish, what we call in vitro human avatars. Those in vitro human avatars capture the diversity of the population by a combination of collecting samples from diverse patients, further modulating them potentially using genetic engineering via technologies such as CRISPR to introduce additional mutations that we know to be or believe to be drivers or protecting of disease. And then those cells are differentiated into the appropriate lineage. So hepatocytes, for example, in the context of liver, or neurons if you're dealing with a CNS disease. And then these cells are measured using very high content readouts based on which we then generate those machine learning manifolds that help inform our understanding of patient heterogeneity and the structure of the patient population. But what's important here is that this is also an intervenable platform that allows us to do experiments in ways that we would never be able to do on a human population. So we can basically screen this population for what happens when you intervene at a particular gene or with a particular drug. So let me demonstrate what this looks like. This is not work that we did. This is work that was done by a group at UCSF on a really interesting genetic aberration that exists in a subset of humans. It's a locus on chromosome 16 called 16p11.2, which in some people is that entire region is duplicated, and in other people that region is deleted. So it turns out that in the deletion patients, there is a 75% penetrance of autism, and in the duplicated individuals, there is a 40% penetrance of schizophrenia. But we don't really know what in the region causes this particular yin-yang phenotype to manifest. This group at UCSF took samples from patients with the deletion and with the duplication. These cells, they took these cells, reverted them to stem cell status and differentiated them into neurons, and then looked at them under a microscope. And if these are the wild type neurons, what you see here is the autism patients or the deletion patients, and what you see here are the duplication patients. And even to the untrained eye, you can see that these neurons are very different in terms of the soma size, the size of the center of the cell, as well as the axonal aberration. And that can actually be quantitated using computational methods, and the differences are very statistically significant. So you can think of this as creating now a way of measuring cells from different patients that allows us to potentially build this manifold that we talked about, hopefully using more quantitative, much richer ways of measuring those cells, and then look for interventions that might revert some of those aberrant phenotypes back to the wild type. So this was work that other people did. We're applying this approach to diseases of the CNS, for example, like Parkinson's disease, where if you think about the very broad spectrum of genetic drivers that induce Parkinson's disease in different combinations, leading at the end of the day to the death of midbrain dopaminergic neurons. That process goes through a bunch of very different pathways corresponding to dysfunction of mitochondria, these little energy factories that we have in the cell, or impaired autophagy, which is the ability of cells to clean up protein messes that exist in those cells, defunct proteins that are clogging up the cell. The combination of these pathways is present to different extents in different patients, and by looking at the cells using these high content methodologies, we're able to disentangle those processes and figure out what are the clusters of patients and how to treat each of them in ways that are very difficult to do from human clinical data, because by the time you get post-mortem brain samples, most of the dopaminergic neurons have actually died. So with this intervenable platform, we're able to test genetic interventions, which are potential ways of taking a cluster of patients and reverting it from a sick state to a healthy state. Those are potentially novel targets. We can also test out compounds to see if those end up reverting the phenotype from a sick cell to a healthy cell. And as we talked about our last effort on therapeutic design, it also allows us to use this as a closed loop optimization platform to optimize molecules towards the goal of reverting the phenotype from an unhealthy to a healthy state. So think of it as almost doing reinforcement learning on a biological system. So in order to do this, we've built out an incredible automation capability that allows us to culture and measure stem cells at a very large scale. So this is what our wet lab looks like. You can see that this is our, we can take the cells out of the freezer, prep them, put them on this very high-end automated cell culturing automation system called Vantage. The cells grow, they differentiate, and then they're taken and perturbed on a device that is called the LabSite Echo, which is an incredible device because it allows you to intervene in the media without ever having pipette touch the medium by using acoustics to move cells, to move the culture around, and then you can take the cells and image them using our fluorescent microscope that is integrated with a culturing system that allows that the cells to be alive as they're waiting to be imaged on the microscope. So to give you an example of one of the projects that we've done, and continuing to use NASH as the example here, this was work that we did on hepatocytes. This was before our hepatocyte differentiation protocol was up and running. So we used primary hepatocytes from patients and controls and asked whether you could see the difference between NASH patients and the matched controls by looking at those cells. A person was not able to do that, and so we asked the machine learning to try and distinguish between the patients and the controls. So this was a relatively small data set from a human perspective. We had only 9 patients and 9 controls for training, and 3 for testing. But you can see that on the training set, despite the fact that a human is not able to distinguish between those 2 cells, a machine was quite nicely able to separate out those 2 cells on the training set, and similarly on the test set or validation set, which was not used in training. Now, in this case, our goal is to derive novel biological insights from this analysis, and therefore explainability of the model turns out to be an important aspect. And so we derived several different ways in which we can obtain explainability to convey to a biologist what it is that the machine is picking up on that the person was not able to discern. And so one of the simplest ways to provide explainability here is simply to show a ranking of the different tiles relative to how NASH-like they are versus how control-like they are. And when you look at those, and we have also come up with ways to quantitate that, what we found is that, and first of all, sorry, let me explain what we're seeing here. The pink is the nucleus, the blue is the cell membrane, and the green are those lipid droplets that cause the hepatocytes to become inflamed and rupture. And what turns out to be the case is that the difference between NASH and control is not the total amount of lipid that has been absorbed into the cell, nor is it the number of lipid droplets. It's the existence of large lipid droplets, large green blobs at the nuclear membrane that causes the disruption. And that aligns very well with findings that came out of the genetics, and so has led us to one of the paths that we're now using to prosecute the disease. Let me show you a different example that does use some of our work on stem cells. This is a disease that is a rare disease, but not ultra rare. It exists in about 1 in 6,000 live births. The disease is called tuberous sclerosis, and it's caused by a loss of function mutation in one of 2 genes in a very highly conserved pathway called mTOR. The genes are called TSC1 or TSC2, and that loss of function leads to benign, not metastatic, but quite problematic tumors that preferentially present in skin, brain, and kidneys. Now, the most significant cause of morbidity in this disease is neurologic impairment, which is tumors that grow in the brain. And for patients that have those tumors, epilepsy develops in 90% of them, often within the first year of life, and 1/3 of them have refractory epilepsy that is simply intractable to other forms of treatment. The only drug that's been approved is something called everolimus, which is a rapalog, an analog of rapamycin, which is a very famous drug, and it works in about 50% of patients, but has issues with a number of aspects, including some tolerability effects, as well as blood-brain barrier permeability of the drug. So we wanted to take on and see whether our platform was able to elucidate more of the biology of the disease and identify potentially novel targets. And so this is work that we did on an iPSC-derived model of the disease. Here we use what is called an isogenic set of lines, which is a wild-type line shown on the right and a double knockout that was generated via CRISPR shown on the left. Again, this is a place where you show this to us developmental biologists and neuroscientists, and they're like, these two look the same. There's no way that we would be able to distinguish between the two by eye only. On the other hand, when you see the machine learning in the middle was able to quite nicely separate between the two, giving us the ability to screen towards potential reversion of that phenotype. So this creates a very interesting question of what does reversion look like in this manifold that we've generated, and so how do we test whether something is a true reversion, a partial reversion, or just an orthogonal treatment? So in order to test that out, and this is a very, very early work, we took two compounds that are known to be effective treatments, at least if you avoid some of the blood-brain barrier issues, which is not an issue in culture, the everolimus and rapamycin. We took two orthogonal treatments that are both known to be active neurological compounds, but don't act against this pathway. And so what you see here are the two, so you see the pink dots and the green dots are the baseline healthy and baseline sick, and then laid on top of them are the blue dots, which are the knockouts with the drug, and the orange, which are the wild type with the drug. And you can see that there's no real reversion in either of those two compounds, using either of those two compounds between the sick and the healthy. In fact, if anything, what you see is that at very high doses, which is towards the right, you see some movement from the healthy cells to the sick cells, probably because these compounds are toxic at high concentrations. Conversely, when you start to put in the compounds that we know to be effective, like everolimus and rapamycin, you get partial reversion for some of the cells, you get complete reversion for other cells, and so you can see that this actually does create the ability to screen for effective compounds using this system. So we've talked about, so as a reminder, we talked about machine learning enabled prediction across the R&D value chain. We talked about the work that we've done on target identification, and we demonstrated that using our machine learning enabled statistical genetics, as well as on some of the iPSC models that we've shown. We also demonstrated the fact that the machine learning is able to extract interesting clinical endpoints in the histopathology data, for example, which potentially are able to better empower our ability to conduct clinical trials by giving a very robust, reliable measurements of human clinical state. What I'm going to tell you about now is some of our work on the middle part of this, which is multi-parameter compound optimization using machine learning and active learning. So a little bit of an introduction to to the current state of drug design. The way this is currently done, and this is a broad caricature, so just for exposition, is a human begins to look at the protein structure that gives rise to an initial design of some number of compounds. This is called structure-based drug discovery. Sometimes that is instead done by high-throughput screening of an existing compound collection. But once those compounds are designed, they are synthesized, which is a very expensive and time-consuming operation, tested via a range of in vitro assays. A human sits and looks at those data by eye, and that gives rise to the next stage of design. We're looking to upend that and do it in a very different way, which is to use very large amounts of compounds in order to build a machine learning model that predicts binding affinity of compounds to a protein as well as other relevant molecular properties. We can then go to large compound collections that are available for purchase, often in a matter of a week or two, you can have compounds that show up on your doorstep. Those can then be rapidly tested using the high content phenotypic assays that we developed in our other work. And then those data points are input into the next round of machine learning that can help us build better predictive models and select the next generation of compounds for testing, which again, importantly, avoids the very slow step of compound synthesis, which can often take multiple months. So in order to generate the right amount of data at the scale and quality that we need, we turn to a relatively new technology, one that's very powerful, which is DNA encoded libraries. So this is a cartoon of what DNA encoded libraries are. So maybe jumping to the end and then I'll describe the process. A DNA encoded library is a single test tube that has 100 million, sometimes as many as 1 billion compounds, all mixed together with multiple copies of each compound, each attached to this DNA barcode that tells us what that molecule is. So it's an identifier, if you will, for the compound that exists in the collection. This is generated via a process called combinatorial synthesis, in which the DNA tag is attached to a small building block. You have multiple different small building blocks, also called synthons. They are pooled together in a single pool, and then separated out into a bunch of new pools where a different building block is attached in each of the pools. So, for example, in well A, you have a mix of all of the different pools, and the and the purple triangle is attached to it, whereas in well B, all of those compounds would have the yellow triangle attached to it. What happens in this process is that as the compounds are mixed together, the building blocks are mixed together, they attach to each other, but the DNA barcodes also attach to each other, which allows the barcoding to be maintained as this combinatorial synthesis process takes place. With that mix of compounds in a single pool, you can take your protein target of choice and incubate it with all of those potential compounds together, and then pull it out. It's usually attached to a solid support like a magnetic bead, and with it come a bunch of compounds that are attached that tell us basically what bound and what didn't bind to the compound, to the molecule, the protein, sorry. In most contexts, this type of approach is used as a way of generating starting points for medicinal chemistry. So we use this as a substitute for high throughput screening, and then that's just the starting point for medicinal chemistry. What we do was very much also supported by this work that was done in parallel to us by a group from Google Applied Sciences and XChem. What this group did was they took, instead of taking just the top compounds, they took the entirety of the data set and trained both classic random forest and deep learning graph convolutional neural network machine learning models on those DNA encoded library data sets for 3 proteins, and then they used those to screen these large virtual libraries. And what's notable is they had a pretty high overall hit rate in terms of compounds that were then acquired from those virtual libraries and tested, and they even discovered some very potent compounds. These are what's less than 10 nanomolars. And you can see that in the diagrams at the bottom, where you can see also that there were more high potency hits discovered by the graph convolutional neural networks than by the random forest across all 3 of these proteins. But the hit rate was actually pretty significant on both of these. What's also important from a drug discovery perspective is the diversity of the compounds that were uncovered. Only 2 to 3% of the chemotypes that came out of this virtual library had previously been reported as binders to anything in a large database called Kemble, and in a definition of something called Merkle scaffolds, which defines sort of the high level structure, if you will, of a compound, there were several hundred scaffolds for at least one of the targets. And the diversity is important because oftentimes when you have only one starting point, your molecular design process hits a dead end because you're unable to get that compound to be soluble, gut permeable, blood-brain barrier permeable, one of the many other properties that you need in order to make an effective drug. The more starting points you have, the better off you are in terms of having more probability of success. So we built on top of this work from Google and XChem, but also worked on this, as I said, in using our own way. So the standard approach in this area is basically classification using random forest or discretized graph convolutional neural network. You dichotomize the data into binders and non-binders, which has the effect of removing some of the experimental noise in what is a pretty noisy assay, but also loses all of the quantitative aspects of the signal. So in order to really try and maximize the benefit of these models, we created a pipeline that aims to identify the best labeling strategy on a validation set, train the models using that best labeling strategy, and then, of course, applies the model to an independent test set. We found first that graph neural networks are actually better able than random forest to use noisy training data. So creating looser thresholds then is actually beneficial to the graph neural network in ways that is not so much for the random forest. The other thing that we did is also tried out regression data in which we don't dichotomize the data, but really try to find something that matches the counts, the trains towards the quantitative counts. That avoids a somewhat arbitrary selection of the threshold and allows us to make use of the entirety of the data set in much better ways. So fortunately, we had access to a proprietary data set that we were able to obtain via collaboration. And in that data set, with a very challenging protein target, they also had measurements of, for some of the compounds in the DEL, of what's called a predicted PKD, which is a predicted binding affinity, which is known to correlate with a true binding affinity. And that was a very useful way for us to estimate the quality of our predictions, because ultimately our goal here is to predict binding affinity. And the correlation with the estimated PKD of a model, it indicates its ability to differentiate between strong and weak binders, which is a very important aspect when you're trying to do drug discovery and balance between different properties that you care about. And so we see by looking at the graphs here, where what you see in the bar chart is Spearman correlation, that the graph neural network classification model considerably outperforms the random forest classification in estimating PKD, and the GNN regression, which is our model, is much better yet. We also see that the regression GNN actually has a very strong correlation with the predicted PKD, and that when you're looking for a recall of high PKD compounds, the regression model does considerably better than the classification model at retrieving those compounds. So this was with a traditional DEL approach, the work that we did so far. In order to really up-level our work, we acquired in October of last year a company called Haystack Sciences that has a pretty unique technology stack that helps empower our machine learning approach. So the two technologies that they have that we got really excited about was, first of all, is programmable DEL synthesis. So rather than having the DNA barcode indicate solely a record of the synthesis steps that were taken, here the DNA barcode is a little programming construct that actually tells the synthesis how to happen. So you can basically write DNA that says, I want this building block with this building block with this building block, and then you have this little routing chemistry chemistry that I'll show you on a subsequent slide that generates the relevant building block, that generates a molecule from the relevant building block, which allows libraries of hundreds of millions of compounds to be synthesized on spec, which when you think about this from an active learning perspective, this is actually quite an exciting ability. So, so what happens is that you can basically create a set of building blocks, each with their attached DNA barcode, and then you create a set of building blocks, each associated with a given barcode or codon, and then you can create this little program that says, put this together with this together with this. You can introduce restrictions, such as don't put this with that one, don't put too many of these types of building blocks together because they're bad, like things with rotatable bonds. And so this is effectively a way to design a really great library with computational specifications. That library is then, the DNA pool is then put into what we call these little mechanical ribosomes that basically execute the sequence of instructions to generate that pool that we talked about. So it's a real enabler to machine learning enabled design. The second thing that we were able to get from this acquisition is a technology called Indexer, which aligns incredibly well with our work on regression because it provides a much more quantitative selection, a much more quantitative readout of the binding affinity of the resulting compound than what you get from the simple experiment that I described earlier where you pull stuff out. So it provides much better signal to noise, better sensitivity, and higher dimensional, more robust readout, which is really the right fodder for our regression-based GNN models. So what we then plan to do is to really create this active learning loop in which we start out with an indexer assay for a given protein. You can see the quality of the binding affinities. You can train a regression GNN. You can then put that together with other properties that you care about beyond selectivity, like those drug-like properties. Test those out, and then use the output to retrain, to both rebuild the library perhaps, and then retrain a model that allows the next step of selection to be done, allowing us to go around this loop much, much faster and more effectively. Looking forward to the next step, and I won't go through this in detail, it's a beautiful piece of work done by a group at MIT, Stokes et al., where they demonstrated the ability to train machine learning models towards a phenotypic readout of compounds. This was the readout here was E. coli resistance. They were, so they had a relatively small training set of only about 2,500 compounds with only 120 so-called positives of E. coli resistance. And even with that limited amount of training data, they were able to come up with a model of E. coli of drugs that counteract E. coli resistance, and use that to screen two virtual collections of compounds and identify what is a really exciting molecule, which is a new antibacterial, which is a broad spectrum antibiotic that addresses several antibiotic resistant strains. So our goal is to use a similar approach combined with our iPSC-based disease models to optimize using this kind of closed-loop approach towards a phenotypic output that we have trained to be predictive of human clinical outcome. So really bringing that to those two threads of our iPSC-derived models and this machine learning for molecular design really back together into a single line of work. So, taking a step back, our goal in this effort is really threefold. Like most drug discovery and development enterprises, we hope to discover and develop medicines that are transformative for patients. However, we hope to do it in a very different way, where we really build predictive models for critical steps in the value chain, enabling us to de-risk and accelerate this R&D process, increasing our probability of success and our time to help patients. And importantly, because of the way in which we're building this as a set of reusable platforms that can be repurposed to different uses, this will hopefully allow us to build a flywheel that just keeps, not only is able to do this well and do this quickly, but just keeps getting better and better and faster over time, as is often the case for machine learning models. So with that, I'm going to take the last couple minutes for a big step back and just talk about sort of a little bit of a philosophical note, which is that, which kind of explains why I'm doing this as opposed to the many other things that machine learning people can work on these days. When you take a look at the history of science, you see that in that there are certain epochs where one discipline it made a tremendous amount of progress in a short amount of time because of a new insight or a new way of measuring things or new technology. In the late 1800s, that discipline was chemistry, where we first came up with a periodic table and understood that you really couldn't turn lead into gold, and this is really the birth of modern-day chemistry. The early 1900s is the birth of modern physics, where we understood the connection between energy and matter and between space and time. 1950s is the era where computing came about, where for the first time we were able to build artificial devices that were able to perform calculations that up until that point only people, and sometimes, as we know now, not even people, were able to perform. Then in the 1990s, there were two threads of two disciplines, each of which started to make rapid progress on its own. The first of those is what I call the field of data science, which is related to computing, but is different. It also involves elements of statistics and optimization and neuroscience that really came to form what we think of as modern-day machine learning. The other discipline is the discipline of quantitative biology, whereby a suite of tools, some of which I alluded to at the very beginning of the talk, we're able to suddenly measure biology at unprecedented fidelity and scale and intervene in biology in very precise and scalable ways. And these two fields, with very minimal interaction, kind of evolved in parallel for the last 20 or 30 years. The discipline that I think is emerging as the discipline of the next epoch is what I think of as digital biology. It's the combination of those two last threads come together in a way that allows us to measure biology in unprecedented ways and unprecedented fidelity and scale, interpret what we measure using the tools of modern-day machine learning, and then actionalize what it is that the insights that come out of that by intervening in biological systems using some of the tools that enable us to do that, such as CRISPR or some of the new molecular designs. And so really, that loop is going to transform not only human health, which is the area that we've focused on, but I think it will also transform biomaterials. It will transform agriculture, potentially energy, and many other areas as well. So I think it's a really exciting discipline to be in. It is one that really epitomizes, to my mind, the idea of AI for social good. And I think it's really an incredible place to be. So I hope some of you might become inspired by thinking about the opportunity here and decide to also join on this journey. So thank you all very much.
Thank you, Daphne. It was an awesome talk.
Thank you, Irina.
Apologies for the little mishap in the beginning. It was great to see you. It's been a long time, and yeah, you're getting into new adventures after academia. I think you're kind of moving into different industrial ventures, and this is the most recent one, as I can see. So this is really, really amazing to see that. And I have various questions that I also would like to discuss with you, but I see lots of questions in the Rocket chat, and it's pretty much up to you how we can proceed with that.
Since you see the questions and I don't, I will leave it to you, Irina, to decide.
Right. Yeah.
Questions and audience questions.
Yeah, the audience definitely had lots of questions about different aspects of your talk, and let me maybe raise some of them now. So one of the first questions was, what is your opinion on organs on chips approach versus your iPSC approach?
I think these are different ways of measuring biological systems in an in vitro way, and it's all model systems have their trade-offs. Models on chip tend to recapitulate more different cell types and potentially more complex relationships. They are also challenged by not being as readily scalable, and also generally tend to work for things that are more flat 2D 2D surfaces. An alternative technology that we've played around with more are organoids. Those are self-forming 3D systems that actually begin with an iPS cell and are able to sort of create almost a little micro liver or micro heart with the multitude of cell types that are relevant towards that biology. That doesn't give you the multi-organ aspect that an organ on chip does, but they tend to be generally more scalable and a more faithful recapitulation of that organ on its own. So each of those has its own trade-offs, and each of them are useful in its own way.
Yeah, definitely makes sense. There are many questions also related, of course, to aspects of machine learning applications that you mentioned. And basically, one of them is, for example, is there better correlation of machine learning model prediction compared to pathologists with blood biomarkers due to inclusion of those biomarkers in the training of the model?
No, of course not. This was held out completely. The only thing that we trained is on the pathology data. Both the RNA sequencing and the blood biomarkers were an entirely separate data set that we only use for validation of the model.
Right. And another question is, which stage of the whole drug discovery pipeline, from target identification to and hit discovery to clinical trials, do you think which stage of that could benefit the most from advances in AI and machine learning?
I honestly think that machine learning is going to transform all of those stages over time. Many organizations in big pharma especially started out actually at the tail end of the process, which is the use of machine learning for some clinical trial work, and that's because it's the most immediately beneficial to patients, and also from a, I guess, business perspective. Things that you, given the timelines that we talk about, where the timeline of a drug from the time you begin to work on it until the time it gets into the clinic is years, it's a very long-term endeavor to use machine learning for R&D before it proves itself. And so the use for clinical trials is the thing that came online first. I think the other one that we're starting to see more and more of is the work on molecular design, where there's been an explosion in the number of companies that have done that, partly because there is somewhat more data available for binding affinity than there is for other types of biological problems. And we believe that those data, while useful, are very biased and quite noisy in our experience, which is why we made the decision to invest in our own data generation capabilities using this DNA encoded library company, which we feel will totally enable us to make much, much better binding affinity as well as other predictions relative to just using the data that's out there in PubChem, for example. All that being said, the reason why we made the very significant investment in core biology discovery that is machine learning enabled, because fundamentally the work that you do on identifying the right targets, it has the potential to have the largest impact on our probability of success. The by far largest reason for the many failures that give us the 95% failure rate that we have in clinical trials is not because we couldn't design the right molecules, and it's not because we didn't conduct the clinical trial in the right way. It's because the drug fundamentally goes after the wrong target. And once you go after the wrong target, there's nothing you can do downstream in order to fix that. So we felt like making an investment on the thing that will have the largest downstream impact, even if it takes a long time, is a critical thing for us to spend some of our efforts on, which is why we started there and then expanded into the downstream steps of the value chain.
Thank you. Also questions about basically causal relations that you may expect in the situation. So you mentioned we may expect causal relation from genetic factors to diseases, but aren't there situations where disease is also affected by human behavior, while human behavior is correlated with human genes? For example, when the behavior correlates to some social position and so on and so forth, how do you disentangle such situations?
So those are great questions. So first of all, just to say, one of the reasons why we focus on genetics as a driver of, as a way of discovering causal drivers of disease, because again, at least to a very reasonable first cut approximation, if you see a variant in a gene that is highly associated with a disease phenotype, in most cases, that is because that variation is a driver of the or at least in what's called linkage disequilibrium, which is it is highly correlated with another variant that we might not have measured that is causal of disease. And as we are able to measure larger and larger parts of the human genome and getting to basically whole genome sequencing, the linkage disequilibrium problem, which is the correlation between variants, becomes less of an issue. We have other issues such as multiple hypothesis testing. So that being said, of course, there are confounders, like, for example, genetic variants that are associated with populations and certain population sets, and those have to do with, other behavioral or socioeconomic factors that are more prevalent in some populations than others, and that is indeed a cause for confounders. We try and correct. There's a lot of literature on how to correct, for instance, for population structure as a confounder, for various environmental factors as a confounder. And so there's a lot of ways in which we can help ameliorate the issue of causality. All that being said, ultimately, the most you can hope for from this type of approach is to generate hypotheses that are likely to be correct. And then the fundamental next step is you have to go and test them in the lab, which is why we have a wet lab environment that allows us to test with true causality in a cell-based system. If we modulate this gene, what does it do to cellular phenotypes that or cellular pathways that we know are implicated in the disease? And then, of course, the final proof of the pudding is you put it in a human and you see what happens, and that's called a clinical trial. So everything up until the point that you put this in a human and you see that the human gets better is only confidence building or confidence decreasing, and that's the best you can hope for.
Right. And that kind of also resonates with recent kind of interest in developing more robust models with extracting like invariant features. So this work around causality, which is quite popular in the machine learning field, kind of made me think, like, what is your opinion on potentially applying all the recently developed methods, say, like invariant risk minimization and so on and so forth, that really aim at trying to build models that would be robust to out-of-distribution data sets, that the variability of data due to, I mean, different subjects, different conditions, and so on, will not fool the model, as it often happens in machine learning, because the model will try to essentially extract invariant features rather than spurious correlations. So it seems like your domain is very, could possibly benefit from such methods a lot, but the question is, what is, in your opinion, I mean, what's the situation right now? Are those methods ripe for those applications, or they are still more kind of at the theoretical level being tested on things like Koller dominist? Yeah, so how practical you think they could be?
Yeah, no, that's a great question, and I think as I agree with the statement that this is a very good application domain for this type of technology. I will say we haven't fully explored all of the methods that have been developed for that in that line of work to see which of those might be useful for the work that we're doing. A lot of this, we're very much in the data generation phase, and right now we're happy when we can make good predictions in distribution. But I definitely agree that that's an area that that requires significant work. First, just in order to determine that that you are in fact out of distribution, so that you shouldn't be trusting your predictions. And then how do you learn invariant features so that you can? Right. So I think this is a great area, and I really I think it's something we'll definitely get to, and I encourage people to continue to explore novel methods in this space.
Yeah, I agree with that. I think it's generally a kind of very useful set of potentially practical tools for any kind of medical application, because because of the notion of kind of core invariant things we believe are there in the data, kind of related to certain disorders, for example, or conditions, while on the other hand, you have a bunch of spurious things that you would like to separate from this ones. And
by the way, it's not just in medical data. It also happens on the wet lab side, because no matter how much you care about how careful you are in how you construct your wet lab experiments, there is a tremendous amount of variability across, for example, batches of experiments. Subtle changes in the medium in which the cells are grown can make a big difference. Equipment that measures the assays the cells can drift, like microscopes drift. And so you have out of distribution events that happen all the time, also on the wet lab side, not just on the human clinical side.
Yeah, because it does indeed also relate to this kind of practical question, as you're building this discovery platform using human data, and yet we know that clinical trial populations and other available data are not necessarily representative of the general population, right? And the question is like, how can you avoid possible biases in building this platform and make sure that it's kind of generally applicable? Yeah. So that's that's a good
in
question.
general, I would say, Irina, that this sort of domain is incredibly rich in challenging machine learning questions that can really help push the boundaries of the work that is being done on machine learning, much more than yet another paper on MNIST, which is why I'm hoping that maybe one of the consequences of this talk is that it will encourage people to look at biology as a domain on which, and clinical data on which they can exercise their machine learning capabilities and bring tremendous value to patients, while also pushing forward the state of the art, as I said, much more than another paper on MNIST or ImageNet.
Right. And it also looks like a domain where, I mean, the recent advances with extremely large scale neural nets trained on extremely large amounts of data, kind of, and all the kind of recent interest in scaling laws of deep learning approaches, they seem to be, I guess, in your applications, you, unlike some other medical applications, like say brain imaging, you usually struggle with relatively limited amounts of data. That's why, like, looking at scaling is not quite appropriate. But it sounds like in your domain, you practically you're practically not limited by the amount of data you can eventually collect. So you could look into scaling.
You know, I wish that were true. I mean, there's certainly more of an opportunity to collect very large data sets, especially in terms of the, you know, we're building out this automated infrastructure in the lab for generating large amounts of cellular data. And once that is fully operational, that will become a very rich data production engine. So we're very excited about the opportunities that will open up. That being said, ultimately, we care not about curing cells, we care about curing humans. And so we also need to collect enough human data to align with the cellular data. And that's where, fortunately, there's a tremendous growing recognition in the community that collecting human data in biobanks is an incredibly important endeavor. I mentioned the UK Biobank and all of us. Similar national biobanks are arising in other places as well. Sorry, we seem to have had a little bit of a technical issue. My microphone went out, no idea why, but I apologize for having dropped off in the middle. So not knowing exactly where I dropped off, I'll just maybe close off this answer by saying that to me there's this incredible, exciting opportunity because we're just at the beginning of this wave of digital biology where the tools have been generated and there's others that are coming up, but the really large data collection efforts that make use of those tools to inform our understanding of biology and medicine haven't been deployed as broadly as they can be. And so I would expect that over the next decade, there will be an incredible explosion in large, high-quality data sets that are enabling of our understanding of human disease and our ability to generate therapeutics towards those diseases. So I'm really excited because I think this is the beginning rather than the end of this particular epoch of science.
Yeah, that's great. And actually, that made me also think about the most recent question posted in the Rocket chat regarding exactly the quality of data. So, well, first of all, thanks for the great talk. It seems that one of the major bottlenecks is availability of high quality, translatable, high throughput disease assays. Do you agree that this is a bottleneck? And is there anything machine learning can do to address this particular aspect of drug discovery?
So first of all, I agree entirely that the availability of high quality data that is relevant to human disease or therapeutics design has really been a bottleneck, and we've seen just in the recent advances in AlphaFold, plus the recent work from the University of Washington that achieves a similar level of performance, just how enabling large amounts of data can be, because when we finally started to have enough examples, data samples, where you have a protein sequence and a solved 3D structure, machine learning was able to take those data and turn it into what is by far the best predictive model of 3D protein structure ever constructed by all the biological insight that people were able to bring to bear into this problem. So as we generate large, rich, high-quality data sets towards other problems, I think other problems we hope will also succumb similarly to the use of machine learning. And that's exactly why we built in vitro the way we did. And if you look at the name, in vitro is the combination of in silico and in vitro, and it's the synthesis of the machine learning and the high throughput data production. And we've really brought together some of what we believe to be the most robust and scalable tools that have been designed by bioengineers and cell biologists towards the creation of large data sets that can drive machine learning models, and really designing data sets and building data sets that are fit to purpose towards enabling machine learning to be maximally successful. So I think that's something that one can do across different areas right now. I think the tools will only get better, and hence the data sets that we're able to produce will become larger and better over time. But what I think doesn't work as well is trying to say, well, we're going to sort of take dribs and drabs of data from here and there, experiments that were really never designed for machine learning, with batch effects and artifacts that are almost impossible to deconvolute, and we're going to make a big pile of that, and then say that's a large enough data set that we could apply machine learning to. And what we found over time and time again when applying machine learning to complex data is that modern day machine learning is incredibly powerful at homing in on subtle signal. It is equally powerful at homing in on subtle artifacts. And so if the data set that you have is ridden with artifacts and batch effects and is a combination of these dribs and drabs, it's going to be almost impossible to get the right level of performance from the model, which is why we've made the big investment that we did in in-house data production. Now, to your question on whether machine learning can help enable that, it's interesting that you asked that, because we use machine learning in multiple parts of what we do as we create those data sets. So, for instance, when you're culturing and differentiating stem cells, when a person does it by hand, they get to know their cells. They get to understand that this, certain types of cells need to be treated in one way to be successfully cultured, and other cells might need to be treated differently. This is something that requires many years of experience of a human. We can get machine learning to look at those images of cells using a microscope and figure out what needs to be done to them to maximally enhance their growth, thereby substituting potentially for the hands of a trained technician and enabling us to do this in a much more scalable way. This is just one example of places where machine learning is used not only to interpret the readout of biological experiments, but actually within the inner loop of how we create data in order to improve the quality and the scale of the data that we produce.
Yeah, I think it's actually really a fascinating direction if you think about that, because going back to the ideas we discussed about trying to kind of infer invariant robust models and leave those spuriously correlated noisy things aside, it's currently kind of mainly approached in a passive way, like given the data, try to sort things out. But if indeed you apply some flavors of active learning in order to improve the quality of data you're getting and kind of improve the robustness of model, that might be even more kind of effective. So I'm pretty sure, yeah, as you said, the answer to the question is quite positive, that machine learning can help improve eventually quality of data collected. And yeah, perhaps that's kind of one of the directions to go. There is also interesting question that just popped up. Yeah, from Marina. Thanks again for all the insights. Philosophical and possibly open question. Obviously, there are great successes ahead for digital biology, but did you get a sense of the limits? There is a theory of the learnable. Is anyone asking what biological processes are learnable?
Ooh, that is a very philosophical question, Marina.
It's Marina's
Yeah. No. I said, Marina. It's a great question. I have to admit, I haven't thought about that. I guess what I would say is that learnable is a spectrum. It's not a binary thing. And we all know that the machine learning models that we have today for other problems, they're not perfect. It's not that they labeled, it's not that that the machine learning is able to 100% accurately label sentiment and sentiment analysis or recognize images from the web. So we've not achieved perfection on any of other problems, or in some cases we haven't even achieved human level performance in those areas. So what I would say is it can we learn enough to make this useful, and can we do it better than is being done today? And when you think about the statistics of the success rate of from the time that you enter phase 1 clinical trials until a drug is approved is, as I said, depending on how you count, 5 to 10%. There's an awful lot of room for improvement on that number. Let's just think about, let's just take 7% as a number here. If you go from 7% to 14%, you've doubled R&D productivity relative to where we are today. So I think that's really the thing that we would strive for. It's not perfect learnability, but usefulness.
Yeah, so indeed, it's always a good philosophical question with any applications of machine learning, I think, in general. But yeah, at least, as I say, the proof of the pudding is in eating, if it's useful. At least, you can validate that. But the good question, I mean, again, I think I'm quite interested lately on this invariant kind of model discovery and so on. It's also a good philosophical question. Yeah, what type of natural laws you could, in principle, even hope to discover with machine learning, and which things, I mean, basically where the limitations are. And that's a good question applicable to everything, including, but not limited to biology.
Right.
Yeah. So, but yeah, I think it's really fascinating, but at least it's also good that, I mean, you have immediate practical benefits of that, which kind of also leads to some of the questions people often wonder about when they see a person from academia moving to career in industry. Well, in your case, obviously, I have a long and extremely successful career in academia, and then transition to industry started happening with Coursera and so on and so forth. So, yeah, people often wonder in such situation, like what kind of induces you to make the move, and what are the most challenging aspects of this type of transition?
That's a great question. So I think in terms of what induced me to make the move, it was primarily the challenge of making an impact from within an academic environment on the lives of actual people. So of course you have an impact on the lives of your students, and you train them well, and you hope they go off and do something great. You can write papers and hope that people read those papers and do something impactful with those. But that's all kind of a little bit like, casting your bread upon the waters and hoping that something happens, to use a biblical metaphor. And that is, it felt at some point too arm's length and uncertain. And I wanted to really be in a position where I could make sure that things did in fact get translated into practical utility. And maybe one specific case in point, which is was in some ways sort of part of the thought process, was that one of the last papers that I wrote while I was still an academic was with my former PhD student, Andy Beck, who is an MD PhD pathologist. And we wrote one of the first machine learning for digital pathology papers. This was back in 2010, 2011. And there were a lot of really, I mean, from a machine learning perspective, it was a very simple paper and has since been subsumed by much, much better methods, of course. But it was very impactful as being one of the first of its kind. And I tried really hard, as did Andy, from our respective academic positions. I at Stanford, Andy went on to be a faculty member at Harvard, and we both tried to get those methods used to improve the standard of care in pathology, because we knew even back then that you could do much better with machine learning on pathology slides than most pathologists can do. And we were just not able to get traction for that from within an academic environment, because in order to get adoption, you actually need to build a product that someone can use. It's not enough to write a paper. And honestly, building products is not what academic institutions are designed to do. It's completely misaligned with the incentive structure of a graduate student to productionize their work, because from their perspective, the best thing is to go and write the next paper so they have a good list of publications and they can go and get an academic job. And so to really build something requires a dedication and a willingness to work as a team and not worried about who's going to be first author in the paper and all that stuff that is just not aligned with the incentives of an academic environment. So I felt like if I really wanted to make something happen in the real world directly, I needed to go out and be part of doing that myself. So that was the rationale, and I think it's been quite a journey, and I've learned a tremendous amount. So as to what the challenges are, it is difficult to move from an academic role to an industry role. It's a very different kind of environment. There's much more focus on goal settings and timelines and deliverables. You can't just go off on a tangent most of the time to explore a new and interesting direction just because you happen to think about it. You really need to execute to achieve a certain level of completion. And I think that's a really, it's a discipline that's hard to learn for those of us who are trained as academics. There's also the sort of challenges and opportunities of working as a team, where it really is a group of equals coming together, each contributing in their own way. And again, that's not something that as often happens in academia, where it's really much more of a, you have a lead author who's taking on the majority of the work with some help perhaps from others in many cases, not every case. And learning how to work effectively as part of a team, I think, is a really important discipline. And if you're looking to achieve great things, especially in a domain as interdisciplinary as the one that I'm currently in, it takes a village. It's not something that any single person or even people from a single discipline can do on their own. And so learning how to bring out the best of the different types of people that you have on the team and really get to the point where the whole is bigger than the sum of the parts is also far from trivial and a really important thing to do.
Well, thank you for a great answer, and thank you for a very inspiring talk. And I really hope that your talk will inspire many young people to indeed maybe try to both pursue their academic kind of careers and also to try to do something practical in the world, whether at the same time or sequentially. But I think, it's really great to expose people to these different aspects related to research, from practical, from theoretical, from academic, and from industrial kind of perspectives. So basically, I think that the best suggestion for them is indeed to choose what at a particular time in their life is best for them. And I think your journey is truly inspiring. And thank you for amazing talk once again. I think we're getting close to the end of the session. And I don't know if you'd like to make any final comments or kind of any message to the audience. Yeah, so that would be great. And then probably we should start. So
maybe just reiterating and amplifying your call to action, Irina, which is, this is an incredible domain for people who are interested in not only doing cutting edge machine learning, because I can tell you that the problems that we are working on really require machine learning out of the box is not what you usually need here. You really need to understand the problem and then figure out how the broad toolkit of machine learning can really be applied towards those problems. And importantly, it's not just a challenge problem for machine learning. It's truly, as I said, a sort of a flagship problem, if you will, for those people who are interested in AI and machine learning for social good. There's a lot of problems, of course, where machine learning can be deployed effectively, and some of them are more beneficial to humanity than others. This is, to my mind, on the high end of things that can really make a difference to the lives of people. So for those of you in the audience who care about that, I would encourage you to consider this space as one of the areas in which you could deploy the toolkit that machine learning has developed for us, because it is really an inflection point, and smart machine learning people getting into this space and making a difference, I think is going to be transformative to the discipline and to humanity. So think about that as one of the options as you think about what you want to do with your career.
Thank you, Daphne, for great advice. And thanks, everyone, for listening, and thanks, the organizers. I guess on this note, we're concluding this session.
Thank you.
Thank you all.
Thanks, everyone. Thanks, Irina and Marina and Tong. Thank you. Bye.